Second, to introduce our speaker today, Yige Zhang, who comes to us from Texas A&M University, which is one of my alma maters. And Yige did his PhD at Yale and a postdoc at Harvard, so not bad schools there. He actually did his postdoc with Ann Pearson, who was here a couple weeks ago. And I got lots of fun stories about Ann last night during dinner, so that's cool. Yige is a paleoceanographer and an organic geochemist. He's interested in a whole slew of things that have to do with the ocean, including CO2, circulation of the ocean, methane. Today, he's actually going to talk to us about cycling of greenhouse gases in terms of CO2 and methane. Very recently, Yige was awarded by the Chemical Society the F.W. Clark Medal, which is given to a early career scientist who, for a singular contribution to geochemistry. So that's quite a big honor. And the other thing I learned about Yige last night was that he's a fisherman extraordinaire. And so we spent a lot of time talking about fishing, and I learned all about a brand new fish that I'd never heard of called ribbon fish, which I now would like to eat. Without further ado, take it away. Thank you.
Thank you. Thank you, Dan. Thanks, Maggie. Okay, everybody, for having me here. Just for your information, I tried to go to the bathroom right before this. I tried all 3 floors, they are all locked. So if I sounded grumpy or that might be the reason why. But I think I'm going to do fine. And, I will talk about organic geochemistry and its constraints on 2 greenhouse gases, CO2 and methane, in my presentation. I think all of you guys have heard a lot about organic geochemistry, and hopefully I'm going to demonstrate that organic geochemistry has a lot to do with either the variability, reconstructing the changes of CO2, or the cycling, the biogeochemical cycling of methane. So that's what I'm going to do. I love both of them, CO2 and methane, but because I only have 15 minutes or so, I can only, I guess, focus really on one. So I'm going to do methane in this case because, like Dan was saying, Anne was here, and I think she was talking about CO2. So we're going to get a methane covered as well. So that's what we're going to do today. Okay. So when we look at global warming, we look at climate change, we look at greenhouse gases. It's important to distinguish the non-condensing greenhouse gases, right? Because if we look at water, yes, in terms of its not radiative effect, it is important. But the thing is that water has a lifespan in the atmosphere for hours, days, weeks, right? But these two are related to the carbon cycle. So carbon cycle operates on different time scale, short time, long time can be as long as thousands, tens of thousands, or hundreds of thousands of years. So once these gases are in the atmosphere, particularly CO2, they're going to stay there for a long time. So that's why we consider them as driving forces. And you don't just increase CO2 by like warming up the ocean, that kind of thing. But that's the opposite of water. So that's why we're going to look at those two, and we're going to look at the organic geochemistry perspective. Okay, so talking about CO2, we all have seen these iconic, this is the Ewing curve, right? This is the instrumental recorded CO2 variations, and you see it increases monotonically. So one thing that people can do is that instead of telling people which year you were born, you can tell people what was the CO2 level that you were born. For me, it's 344. So if you're curious about when I was born, that's 344. But if you keep going back, you see that's the ice core recorded CO2. And very nicely, we're looking at the glacial, and that's the last glacial maximum. That's the penultimate interglacial. We're looking at these glacial interglacial variations. So largely changes between 180 to 280 ppm. Versus today, we're at 410. 415 ppm. So that's way above the natural variability over the late Pleistocene glacial interglacial timescale. So if we want to find an analog of that, 410 ppm, we have to go very far back in time. And that's when we have to rely on geochemical processes. Ice does not go back that far. You know, you might have heard people are hunting for these very old ice, they call it blue ice, but that's like 1 million years or so. But beyond that, we need geochemical or paleontological proxies. So one of the first and perhaps one of the most famous proxy for atmospheric CO2 is called the alkyl CO2 method. And this is based on organic compounds called alkyls, and they're produced by marine phytoplankton and coprolithophores in this case. And this is based on the concept that during photosynthesis of these algae, they fractionate carbon, right? And they fractionate carbon according to the concentration of aqueous CO2 and other parameters like their growth rate and their physiology. So if we do this well, then we can use the isotopic fractionation and calculate aqueous and atmospheric CO2 level. So I listed a bunch of like very early papers. You see Kate's name here. A lot of people coming from John Hayes' lab, who was Kate's advisor. And it's also, it's nice to review papers. And the connection here is Pagani was my PhD advisor, who was Kate's student. So that's the alkyl CO2 method. And it is developed in tandem with the technological advances, and that is compound-specific isotope analysis. You know, before we're able to measure isotopes on bulk, we're able to measure it on carbonates, but back in the late 80s and early 90s, I guess that's the very exciting time in John Hayes' lab, where, you know, people developed this technique for which we can measure isotopes on a single compound, right? So very briefly, it works in the way that we use a gas chromatography to separate different compounds, and we combust them, we convert them to CO2, and that CO2 goes to a mass spec. So that's how we learn about their carbon and their oxygen isotopes. And eventually that goes to a computer that's a compact 386. It's
You
cute.
thought that was pretty snappy.
Yep. I can imagine that. So yeah, that really opened up a door for geochemists to look at these compounds. You know, we get much fancier today. You know, we have these site-specific isotope. You know, that's something I guess Maggie or Kate, you could talk more about. But it all began here. This is when we're able to look at single compound for their isotopic composition. So one of the first applications is actually alkyl CO2. So that's what we look at. And this approach has led to a very important discoveries. So this is a paper by Mark and Jim Zakos, Kate, and other folks in 2005. And it demonstrated over the Cenozoic timescale, CO2 experienced this major decline. And if you look at the greenhouse world of the early Cenozoic, which is the Eocene, you see that CO2 level was on the order of thousands of ppm. And then it declined very largely during the Oligocene and transitioned into the Neogene cool house, the ice house world. Well, that's when we had CO2 on the orders of a few hundred ppm. So that's on par with what is happening today, right? So that led to these discoveries that carbon dioxide in the atmosphere is declining over the Cenozoic timescale, and that's leading to this major Cenozoic cooling. And this is 2005. This paper is cited thousands of times, but you know, over the recent years, we also realized that all proxies have its limits, and proxies has its own issues. And I guess over the recent years, people are debating about how to make the alkyl method better. I think it's fair to say that I think the key is to understand physiology, because I mentioned that these things are done by algae. So they are biology, right? There's no reason for them to only respond to CO2. They do their own things. So they have their different growth rate. They have potentially, they use a carbon concentrating mechanism. They actively pump carbon instead of passively diffusing carbon. There's other issues like light availability, nutrient, all kinds of physiology that complicates the problem. So I spent the last quite a few years, actually for my for my postdoc, and for when I started my faculty position, I've been working with my student, particularly Xiaqing Liu, on how to refine this approach. And we realized that algal physiology parameters and their relationships, they're important, but they're super complicated. Okay, for very simply, like from the size of the cell, you want to calculate the amount of carbon of that cell. It can give you 12 equations. That's based on laboratory cultures. That's based on marine in situ sampling. That's based on different algae. Which one should we use? Okay, that has been challenging. And that's when we started to learn about these modern advanced statistical analysis. And what recently we have been doing is to use Bayesian statistical model to help provide constraints, the probability density function of these parameters. And ultimately, we want to test this updated method using ice core, because that's the available CO2 changes over interglacial time scale from the ice core. And any proxy, I think it should be compared with the ice core records. So for two things, one, it's the standard, it's the golden standard that we know from the ice. And second, if we demonstrate that the method can resolve this relatively small CO2 changes, this is only 80 to 100 ppm, then we are going to be much more confident when we look at the presumably the larger changes in Earth's history. So that's where we're heading to. That's where we're going. So with that, I would like to switch gears to methane. I'm going to talk about methane. Okay. When we talk about methane, everybody knows there's no proxy for atmospheric methane, right? There's no proxy for how much methane we had in Earth's history. There's a proxy for CO2, alkyl CO2 I just mentioned. There are other methods as well, but there's no proxy. So if you can come up with a proxy, that would be a major breakthrough. But even if we don't have one, it doesn't prevent us from understanding the biogeochemical cycle of methane, right? And in the modern system, near Earth's surface system, we know that there are huge amounts of methane trapped under the seafloor. They're in the form of marine gas hydrates. They're ice-like crystals, so there's methane, there's water in there. And there's thousands of gigatons of methane stored in this way. And for some people, they think that it could be the future of cleaner energy. I mean, it's not really clean, but it's better than petroleum or oil because there's so much of them. But the other peoples are like, no, don't touch them. They're dangerous. They are only stable under dedicated temperature and pressure condition. So if you pull them up from the bottom of the seafloor, they're going to decompose. They're going to release methane, a potent greenhouse gas. So talking about the potential climatic impact of methane hydrates, I guess the most famous example comes from the Paleocene-Eocene thermal maximum, the PETM, that happened about 56 million years ago. And if we look at the PETM, then you see this is marine records. You see the carbon isotope, there's a major negative excursion. So you know there's a light carbon and it has been emitted to the ocean and atmosphere. You look at O18, then you see this, again, negative excursion, that means rapid warming, okay? And if you look at carbonate content in these marine sites, you see before or after this event, these places have 90% of carbonates, but right during this event, this number dropped to almost to zero. So that's ocean acidification. So warming, light carbon, and ocean acidification. I think that rings a bell. That sounds like what we're doing today, right? So that's why people are super curious about what exactly happened during the PETM. And where does that carbon come from? That gave us that negative isotopic excursion. This is a question that bothered people for a long time. And if you look at the C13 of different reservoir, different sources of carbon, then you're gonna see methane has the most negative value. Biogenic on the order, on average of -60, thermogenic a little more enriched than that. But if it is methane, then we need the minimal amount to move that isotopic mass balance. And this is what gave people like Jared Dickens the idea back in 1995 that it is the dissociation of ocean methane hydrate as a cause of isotopic excursion at the PETM. So this is a famous paper and called upon the role of methane hydrates back in 1995. And if you look at what happens after that, you're going to see a lot of papers. This has become like a norm. Like whenever people see rapid warming, they see isotopic excursion, they think of methane. You know, you see people call methane seeps to terminate the neoprotozoan snowball Earth. You see people call methane like carbon injection linked to an Permian mass extinction or mid-Cretaceous warming like CO2 coming from methane hydrates. So a lot of papers like this, right? But the problem is we don't have much evidence. Okay? A lot of these are based on mass balance calculations or carbonate C13, which is subject to diagenesis. Okay? If we look at more recent, we look at the late Quaternary, we actually have this theory called the Clothric-Gunn hypothesis. Okay, we even got a book on this. So this is done by people like Jim Kennett. They look at methane hydrates in Quaternary climate change. And they think the clathrate, the gas hydrate, is a gun that fires every time we had this rapid warming. And their evidence comes from mostly the Santa Barbara Basin record. This is ODP 893, and this is oxygen isotope. Okay, so this is last glacial, this is a Holocene, and this is transitioning into the last glacial. So that's the general changes. And you also see all of these minor spikes. They are the warming events, the interstadials, and they're highlighted by these purple bar. And if you look at this benthic foreign C13, then you're going to see the corresponding negative excursion every single time associated with these warming events. So that's what inspired them. And they were like, oh, this is the methane hydrates in play. And so they emit methane into the atmosphere and warm the planet every single time we have this interstitial. Is that true? Unfortunately, probably not. Because now we're able to look at isotopes, again, isotopes, right? On methane trapped in ice core. So people have done that. This is what people have done in 2006 to 2009. And here, we're measuring C14 on methane, right? And if that deglaciation increase of methane is driven by gas hydrates, then we know the hydrates are old carbon. They should be C14 depleted. They should be C14 dead. So this is what's supposed to happen. Look at this dashed line. If that methane comes from hydrates, this is their measurements. So it's not. And people also have done deuterium isotopes on these methane, also not in favor of gas hydrate source. Instead, they're suggesting maybe it comes from wetlands. Okay, so that's probably the reason why, 10 years ago, 20 years ago, there's a lot of discussions about methane hydrates role. But if you look at the last decade, we don't see many. We don't see many. So that's one reason. The other reason we don't see many that kind of work is because we are making better observations and better modeling of seeps of methane venting from the seafloor. So we better understand where do they go. So this is where they go. See, there's methane coming out from the seafloor and they disappear. They dissolve in seawater. They don't really make it to the atmosphere. The ocean, the seawater is undersaturated for methane, especially under high pressure condition. So they dissipate into the seawater, and except for very, very shallow hydrates, the methane gas does not make it to the atmosphere. Okay, so this is a double hit on like a class rate gun or methane hydrate driven climate change, right? They don't make it to the atmosphere, and the geological records does not suggest that methane come from hydrates. So that's where we're at. And I think it's fair to ask this question. Methane hydrate dissociation driven climate change, is it reality or is it fiction? Okay. And for the rest of my talk, I'm going to answer this question using organic geochemistry. And I think this question boils down to two separate questions. First, what are the right tools to track hydrate dissociation and methane release in Earth's history? First, we have to document. We have to document its history. We have to understand how does it change. And second, even if there's methane emitted, how is it going to affect the global climate if they don't make it to the atmosphere, right? So two critical questions, I guess. Okay, and to answer the first question, you already know the answer. The answer is organic geochemistry. That's not because I'm biased, that's because if you look at methane cycle in the ocean, then you see that it's largely mediated by microbes, right? It's produced oftentimes by microbes and it's consumed, it goes to microbial oxidation. So if we have hydrates in here, in sediment, methane hydrates, and there are two pathways to consume them. One is to consume them anaerobically, that involves the sulfate reduction. And the other way that goes to the water column and where we have oxygen and it's going to be oxidized into CO2. Okay, that's all done by microbes. For anaerobic oxidation AOM, it's done by methanotrophic archaea, the AMI groups, and sulfate reducing bacteria. And they have their diagnostic lipid biomarkers. Okay, these archaea, they have a lot of biomarkers associated with them. For aerobic oxidation, that's done by methanotrophic bacteria, and they also have their biomarkers. They have these hopanoids, and I listed HOPE 1721 in over here. So we can find lipid biomarkers and their stable isotopes to tell if there was anaerobic oxidation or aerobic oxidation happened in Earth's history. And for archaeolipids, anaerobic AOM, we have archaeol, DAGE, we have procetane. For bacterial source, then we have diplopterol, diploptene, and their diagenetic products. And again, this goes to Kate's work. I guess this is when you're doing postdocs, right? Is that your
My PhD.
PhD?
Yeah.
Okay. That's where when they look at these isotopic biogeochemistry to predict the functions, the sources of these compounds. And one particular one of interest here, I like the negative ones. I always like the negative ones. And here the most negative one is the diploptene. So that's the one that I mentioned, diploptene, diplopterol, and their diagenetic products. So they are probably related to methanotrophic bacteria. I mean, they could have a general bacterial sources, but the very negative ones are typically related to methanotrophic bacteria. So that's how we know there's aerobic methane oxidation. Okay, in addition to all of that, okay, we also have these GGGTs. GGGTs are very popular biomarkers. I'm sure a lot of you have heard about it, largely due to the popularity of this proxy, TAX86, right? It's an ocean temperature proxy, and it is archaeolipids, and people use it for ocean temperature reconstructions. But when I was doing my master's degree back in 2011, we look at methane hydrate impacted samples. And this is normal distribution of GGGTs. Even if you're not geochemist, organic geochemist, you know, the size and the, the area of these peaks represent its like relative concentration, right? So in normal marine, we see there's a lot of GGG0 and chronoarchaeol, and these are minor ones. But in hydrate impacted environment, we got a lot of these GGG1, 2, and 3, and we think they come from the methanotrophic, the AMI group archaea. So any group archaea, they're very preferentially synthesizing these 1, 2, 3. So that's how we come up with this thing, we call it the methane index, based on these GGGTs to tell you if there's anaerobic methane oxidation in the sediment. So why do we need this? Why do we need a GGGT-based biomarker, given we already have a lot, right? I think the difference is that people just love to measure GGGTs. There are thousands and thousands of reported GGGT data. So this is a compilation that my student Veronica did. You look at the occurrence of reported GGGTs over time. This is a Mesozoic, this is a Cenozoic. It has very good coverage. And the good thing is that you can calculate methane index at the same time. You don't need additional measurements. So I think that's a good thing about it. And that changed the skin that, if you just want to go anywhere, measure biomarkers and hoping to find methane, that's kind of like, finding a needle in a haystack, right? But because we have thousands and thousands of these measurements, they give us hints about where methane potentially have happened. Okay, so, and I think this is the last one about the method. Just to inform you, the more recent stuff, my student Bangsu Kim and I are working on developing methane index more into a quantitative proxy. So before it's more like the presence or absence of methane. But now we look at the modern system and we see that it's quantitatively related to the flux of methane. It's related to the flux of sulfates. So we look at different boundary conditions. We did the sensitivity analysis. And we think it can be used quantitatively to tell you how much methane was released back in time. So very briefly on that. Okay, so that's all about the method. I hope I convince you that the method, organic geochemistry, is going to rock in terms of informing us about methane history. The next question I ask is, does it matter? Why does it matter if the methane doesn't make it to the atmosphere, right? And the answer is it matters because of these two different pathways, okay? If we're doing it anaerobically, if we're using sulfate, okay, that's related to sulfate reduction and maybe pyrite formation, and it generates bicarbonate, which adds to alkalinity. So oftentimes at places where you have hydrate, you found alphagenic carbonates. There are a lot of alphagenic carbonate platforms in the Gulf of Mexico where we have these hydrates occurrence. And of course, there's a lot of muscle that you see, all of this muscle over here, and there are carbonates as well. So it helps in terms of carbonate precipitation versus if that methane goes to the water, it's going to be oxidized aerobically. And the consequence of that is CO2. That's acid. That's going to consume oxygen. That's going to acidify the ocean. And this has been demonstrated by the Deepwater Horizon incident back in 2010. It's a tragedy, right? Everybody knows that the platform blew up and there's casualty, there's loss of property. But it's also an experiment for people to look at what happened to the huge amount of methane emitted to the water column. And the answer is microbes, they eat them up very rapidly in a few weeks. So you look at this paper, this is the cruises that people did very quickly after Deepwater Horizon incident. And they track the dissolved oxygen in northern Gulf of Mexico, and they see the decrease of oxygen. Oxygen is being used to oxidize methane and petroleum. And if you look at that's utilized dissolved oxygen, and this is the methane concentration. So this is days, and you see the decrease of methane concentration. So that informs us. It uses oxygen, acidifies the ocean. And if this happens massively in Earth's history, it's going to make a difference. So I think this is the key, that really methane is going to make a difference. instead of maybe directly going into the atmosphere. So yeah, that's my answer to these two questions. We're going to use a little bit biomarkers and large scale aerobic oxidation of methane could cause ocean hypoxia and acidification. Okay, now let's move on to one example of this. Did this happen? When did this happen and what did it do? Okay, so we're going to focus on this interval, the Oligocene-Miocene boundary. This is the benthic O18 record, that goes curve. And you see at the Oligocene-Miocene boundary, there's a rapid glaciation that's kind of similar to the prior event, the OI1 glaciation that marked the onset of Antarctic ice sheets. But the strange thing about this IMI1 event is that the glaciation was terminated very rapidly, and O18 bounced right back to almost the pre-event levels. So why? Why didn't this glaciation last much longer? That's a question about a lot of people. And we want to evaluate the potential role of methane in this transient glaciation. Okay, and we look at 3 Southern Ocean sites. There are 3 of them. 2 of them are close to Tasman Sea, and this one is right offshore of Antarctica. So we're going to look at records coming from these 3 sites in the Southern Ocean at about 23 million years ago across the Oligocene to Miocene boundary. Okay, so first I'm showing the background information of these 3 sites. So I have 2 panels here, and you're looking at, this is the Tasman Sea record. That's the O18 record. You see the IMI1 glaciation, and then the warming after that. And this is reconstructed sea level, global sea level, and associated with that glaciation, there's maybe like 50 meters of sea level drop. Okay, and now here on the right hand, that's the Antarctic site. So that's a site U1356, right? Similarly, you see the oxygen isotope, you see the sea level curve. Okay, what I'm going to do now is that I'm going to plot the methane index values of these 3 sites. And remember that the value, if it is higher than 0.3 to 0.5, that's suggestive of methane occurrence and anaerobic oxidation, right? So I'm going to plot them right here. Okay, there we go. So methane index values, background level shoots up, a jump right during the glaciation. Okay, same thing at this Antarctica site. Okay, that happened in 3 different sites and happens right when we had the sea level, low sea level stand. That's when methane shows up in these 3 sites in the Southern Ocean. Okay, and we also did the other biomarkers. Remember, there's a whole bunch of them. There's not just GEGTs and methane index. And we also found there are these compounds, isotopically depleted compounds, that only show up when we have high methane index values. And they're below detection when it's normal methane index values. Okay, and there's some indication of not only anaerobic, but also aerobic methane oxidation. So maybe that methane vented into the water column and caused an aerobic oxidation. So yeah, 3 sites. I don't think it's a coincidence. Instead, we think maybe that's a sea level driver that caused the 3 sites to show up of methane at the same time. And it works the way that when we lower the sea level by 50 meters, you see this is the hydrate stability zone right down here. But if you remove hydrostatic pressure, then you're going to destabilize this wedge of the GHSZ. And that's going to cause methane to be released in the pore water and maybe also in seawater. And if they're oxidized either anaerobically or aerobically, and that's how we register these lipid biomarkers in the sediments. Okay, so 50 meters, it's not a lot, it's not a whole lot compared to the depth of the water column, right? How much signal are we gonna get? So we did some thermodynamic calculations to show that if it is shallow enough, like 700 meters water depth, and that's actually gonna move up the stability zone by quite a bit, by like 21 meters, and that's 25% of what we have over here. If it's a really deep water, like 2,000 meters, it doesn't matter that much. That's only like 1% changes. And we can also plot it this way. You can look at the different climate zonation because the bottom water matters a little bit, right? The temperature of the bottom water and the percent of this stability zone reduction. If it's a really shallow, right, then we're gonna lose all of it. But as we increase the depth, the percentage that is lost decreases. The other way to look at it is like this. You see the very shallow places, all gone, okay? And relatively shallow, a larger portion, but very deep ones, not much changes, okay? Can we quantify that globally if it's really driven by eustatic sea level? We try to do that. We look at the paleobathymetry of 23 million years ago. We look at the area of different seafloor with different depth, and we look at different climate zonation, different water depth, and eventually, this is what it really matters. We calculated the total amount of methane release in terms of carbon. That's about 200 gigaton. 200 gigaton is not a lot, Like today, we already emitted about 500 gigaton anthropogenic carbon emission. It's not a huge amount, but it's a significant amount, I would say. It would matter. And it would matter in terms of changing seawater chemistry. So we calculated its impact on seawater chemistry with the assumption that the majority of the emitted methane goes to aerobic oxidation. You see that's gonna cause changes in DIC, in C13 of DIC, not a lot to dissolve oxygen. pH changes is about 0.03 unit. And in terms of CO2, assuming CO2 is in equilibrium between the ocean and atmosphere, that's 38 ppm. 38 ppm. Again, it's not a huge number, but I would say it's a significant number. I mean, maybe during deglaciation, we had a couple hundred ppm of CO2 increase. And maybe methane oxidation contributed to 38 ppm of that. So that's what we learned from this oligocene-myosin boundary, the MI1 event. Okay, so very briefly to summarize that, lipid biomarkers are powerful tools to trace past methane release. I think everybody would agree on that. And ocean acidification and hypoxia caused by methane oxidation could be more important than direct emission radiative effect, because it's very difficult to make it happen unless you're in very shallow sea. And lastly, in this case, during MI1, you see that hydrate responded to presumably the sea level drop, but it also potentially drive some sort of climate change. It acted as a negative feedback that perhaps prevented the Earth from further cooling and glaciation. So that's what we see during the MI1 glaciation. So I have 10 more minutes, so I'll just talk a little bit about the holy grail of all methane studies. That would be PETM, right? Was there methane during PETM? The answer is yes. So I'm going to show you the evidence of that. PETM, it's been hypothesized as a catastrophic methane release. But so far, we have not been found good evidence of that. Okay. But in my opinion, what if it's just an enhanced aerobic methane oxidation? It's going to give us the acidification. It's going to give us the CO2. And if that's the case, where would it happen? Where would it happen the most? Okay. I think PETM is a good time to test that because during the PETM, we have low sulfate levels. Okay. So AOM uses sulfate If you look at reconstructed sulfate concentration, if you look at isotope of sulfur during the PETM, they are all indications of low sulfate level and maybe reduced pyrite formation during the PETM. Maybe that's due to low sulfate and AOM doesn't work very well. It cannot suppress the methane during the PETM. So you have to be oxidized aerobically. And if that's going to happen, the canary in the coal mine is the Arctic Ocean. The Arctic Ocean during the PETM look very different from today. Today it's fresh, it's relatively fresh, right? It's shallow, but during the Eocene, during the PETM, it's like a semi-enclosed basin. So it's very fresh water. Other periods of Eocene, we even had these Azolla fossil recorded in the Arctic. You might have seen them in like YouTube, like a lot of nutrient pond, like a local pond. If you got a lot of nutrient, they grow on top of it. It's a freshwater algae, and people have found that fur. Sorry, it's not algae, it's a fur. And people have found that during the Eocene in the Arctic Ocean. So it's a semi-enclosed basin with low salinity, potentially like brackish water. That means we're going to have a very low sulfate level. So sulfate level is low to begin with. And in that particular environment, it can go to sort of extremes. Okay. So if it works that way, then normally you see methane vented from the sediment. A large part of that is going to go to anaerobic oxidation. So that does not make it to the water column. But in the Arctic Ocean, if we have very limited sulfate concentration, then maybe we're going to have a higher flux of methane going into the water column, causing aerobic oxidation. So that's what we think. Okay, now our beloved organic geochemistry, this is GC chromatograms. Okay, I'm showing you the samples from PETM in the Arctic Ocean. Okay, this is the gas chromatography. The chromatograms are from gas chromatography, and a lot of these peaks are compounds. There are a lot of normal LK, and this sample comes from pre-PETM. This is a PETM sample during the onset of PETM in the Arctic Ocean. Okay, this one, if you recall this one, this is the hopanoid that represents aerobic methane oxidation. And it is present pre-PETM to a smaller amount, but look at what happened during the PETM. It became the predominant compound preserved in the sediment. Okay, it showed up in huge amount during the PETM. So, and we also measured its isotopic values, and they're isotopically depleted. Okay, particularly during the PETM and after, but not in the Paleocene. So this is their abundance. This is the carbon isotope excursion that marks the PETM. And we found these compounds in high abundance and they're isotopically depleted. So I think that's suggesting there's some methane venting in the Arctic Ocean and they have been aerobically oxidized. And the consequence of that would be ocean acidification and higher CO2 levels in the atmosphere. I don't think they're the only cause. There's no way this is the only cause of the PETM, but my gut feeling is that it played a role. I mean, this is unpublished. This is not like a well thought out study, but that's what we're thinking currently. So that's about the PETM. And I think this is the very last slide that I have. If you think about this process on the long term, from hundreds of millions of year time scale, right? I mentioned that there are thousands of GDGT data have been published, mostly for the purpose of tax 86. We can use that to calculate for methane index and look at a general picture of how much methane do we have in global marine sediments. Okay, so this is what we did. The red are the anomalously high samples. Okay, the blue are like a borderline. You know, it could have been impacted by methane, maybe not. We can't say for sure. If it's above 0.5, we're almost certain that there was methane. But if it's between 0.3 to 0.5, maybe, maybe not. But we plotted up for the last 120,000 years, 120 million years, right? So this is the Cretaceous, and this is the Cenozoic. And look at the occurrence of these anomalously high samples. Okay, they don't happen much during the Mesozoic, but they peaked during the early Miocene for some reason. Okay, and we also found there's another data. These folks have then compiled the global orthogenic carbonate. Remember that AOM leads to orthogenic carbonates. They compiled the orthogenic carbonate content over the same period, and it also showed up high level during the Miocene. And what is causing that? Assuming this is real, then it could be related to carbon cycle, organic carbon burial, sulfate concentration, sea level, all of these broader boundary condition changes over this extended period. But yeah, this is also unpublished, but I think it informs us that the methane system is changing over time. And it played a role for ocean chemistry, for climate, maybe not through the traditional way people think that they make it to the atmosphere, but through more of its oxidation, its occurrence, its destruction. And there are a lot of stories there waiting for people to explore. Okay, and with that, I would like to thank all of the founding sources, thank all of my students. Thank you all very much.